Some are very simple, some are very complex. Out of these, we are basically, this chapter is basically dedicated to these organic. Organic molecule can be basically of two types, macromolecule and micromolecule. Further, we will discuss, right? So organic molecules are further divided into two parts, right? There are some macromolecules. They are basically larger in size. So on the basis of their size and the size of the molecule or density or weight of the molecule, we discuss, we calculate in Dalton, right? So macromolecule are larger in size. For example, DNA, right? So DNA is a type of macromolecule. It's a bigger molecule. And the second one are micromolecule. So micromolecule are smaller one, right? There may be some sugars and all that, right? So these are basically, out of these organic molecule, it may be protein. So basically we will discuss protein, carbohydrate. So we'll discuss in this chapter carbohydrate in detail, right? Then we'll discuss the fat or lipid. So Iram, can you tell me what is the difference between the fat and lipid? Is there any difference between the fat and lipid?
Yes.
So what is the difference between them? Look, if you ask me what is the chemical difference between them, then there is no chemical. They are not chemically different. But yes, they are a bit different. If when we talk about they are a bit different when we talk about their density, right? Fat is anything which is made up of fatty acid of glycerol. But okay, Aisha, fine. So Aisha, is it your first class? Yeah. Am I audible to you, Aisha? Hello. Okay. So yeah, Aisha, you are not audible to me actually.
Yes.
Okay, fine. So we have just started with the chapter biomolecule. Just we have started with that. Fine. Okay. So let me introduce you that this is the like we have already completed. We have already started the cell biology. Out of this cell biology, we are going through with the biochemistry part, means the chapter biomolecule. And in case if you are facing problem in anywhere, so you have to tell me, right? Fine. So look here. There are the cell is made up of two kinds of component: inorganic component and organic component. Inorganic component are sodium, potassium, or rest of the type of one. Just a minute. Okay, there. Fine. So inorganic component are sodium, potassium, calcium, and rest of the component. And when we talk about the organic component, out of this organic component, there are two types of the thing. First, macromolecule. Macro means the larger in size, and micromolecule means the smaller in size. So size of these macro and micromolecule we measure in Dalton, right? So there are two kinds of the macromolecule and micromolecule. So out of these organic component, the organic component, basically this chapter is dedicated to organic component. Organic component are basically protein, carbohydrate, fat. So I was telling you what is the difference between the fat and lipid. The only difference between the fat and lipid is density, right? Fat have a lower density, and lipid is a greasy kind of the fat. When we talk about the oil, it is a fat, but when we talk about the butter or cheese, it is a lipid, right? So lipid is when fat become denser and it is greasy, it become a greasy like substance, then we call it lipid. Otherwise, there is no difference in the chemical composition of the fat and lipid. Both are basically made up of the fatty acid of glycerol, right? Then in this chapter, we study about the nucleic acid. When we talk about the nucleic acid refers to two things. Either we talk about the DNA or we talk about the RNA. So both have the suffixes deoxyribonucleic acid and ribonucleic acid. So in the both of the term, nucleic acid is have been add on, right? So both are basically called collectively called nucleic acid, right? And last but not the least, we will study here enzyme, right? So these are the basic component of the cell. This is the basic thing which we are going to study. Is it clear?
Yes, sir.
So please start writing now. Put the heading biomolecule. So what are the biomolecules? Please start writing. The chemical or molecules, the chemical or molecules present in the chemicals or molecule present in living organism, present in living organism are known as biomolecule, are known as biomolecule. Full stop. The collection or sum total of the collection or sum total of different type of biomolecule, the collection or sum total of different type of biomolecules, compound and ions present in a cell, present in a cell, present in a cell are called cellular pool. Next point.
Sir, are called
cellular pool. Cellular pool, right? So everything which is present in protoplasm, basically we call the cellular pool, right? All these things, these all organic and inorganic things which are floating inside the cell, inside the plasma membrane, we call that cellular pool. Suppose that mitochondria want something from outside, like it want to make the ATP, it require phosphate, then phosphate will be taken from the cellular pool. So when we talk about the cellular pool, it means all the chemicals which are present inside the protoplasm are called cellular pool. Is that correct, Aisha, Kiram? Correct to both of you?
Yes, sir.
Okay, right. Next one. The biomolecules are of two types. The biomolecules are of two types: inorganic and organic. Inorganic and organic. Full stop. Inorganic biomolecule are mineral. Inorganic biomolecules are mineral, gases, and water. Mineral, gases, and water. Full stop. Organic constituent. The organic constituent are carbohydrate, lipid. Organic constituent are carbohydrate, lipid, amino acid, protein, enzyme, vitamins, etc. Right? So Now come to the macromolecule and micromolecule, right? So I told you that there are in cellular pool, or rather than cell. So all the component of cellular pool means cellular pool. So cellular pool have two type of the component, or cell is made up of two kind of the component, inorganic and organic. Further, inorganic is like salt, water, and gases. Organic is protein, carbohydrate, fat, etc. Further, the organic molecules have been divided in two categories: macromolecule and micromolecule. So start writing now. The molecules, depending on the size, the molecules depending on the size have been divided into two categories. Have been divided into two categories: macromolecule and micromolecule. Full stop. Next point. The micromolecules are small sized. The micromolecules are small sized, simple chemical, small sized, simple chemical, having low molecular weight, in between 18 to 8,000, 18 to 800 Dalton, 18 to 800 Dalton. So all the molecules which belongs to between this category 18 to 800 Dalton size, we call them micromolecule, right? Next point. In the same point, next point. They have higher solubility. They have higher solubility and simple conformation. They have higher solubility and simple conformation. For example, sugar, amino acid, etc. For example, sugar, amino acid, etc. Next, macromolecule. They are larger in size and complex chemical. They are larger in size. We are talking about the macromolecule. They are larger in size and have complex conformation. Full stop. They have high molecular weight, usually 10,000 Dalton and above. Usually 10,000 Dalton and above.
Sir,
yes,
sir,
yes, I said.
what is Dalton?
Dalton is actually, it is a unit of measurement of the size of any molecule, right? So Dalton have been considered when you talk about the atomic weight, right? So it has been compared with the atomic weight, right? 1/12 of the carbon that is included, that is considered as a 1 Dalton, right? So Dalton is actually this is a size of molecule, right? It's a unit. Dalton is a unit, right? And the unit have been given by, like, unit have been given the name in the respect of the Dalton. You know, the Dalton's concept we study in the class standard 6 or 7, right? So Dalton was the person who given the concept of molecule, right? So the respect of Dalton we have given. He said that the atomic mass of the atomic mass of the hydrogen is 1/6 of the carbon and all that, right? So okay, so they are insoluble, they are insoluble and always inorganic. They are insoluble and always inorganic. Underline always inorganic. It means what? It means micromolecule can be organic as well as inorganic, but macromolecule will always be inorganic, organic, right? Is that clear to both of you?
Yes, sir.
Okay, Aisha,
Yes,
are you
sir.
getting everything? Because it will take some time for you because this is your first class. Fine. Some
Sir,
more. Yeah, please.
can you send me the recordings of last chapter? I was there in the first class, but I missed a few classes in between. So this is my
No
second
issue.
class.
What you can do, I'll say the team as well, right? So, do you have the Learnivio number, Learnivio official number?
Yes, yes.
Right. So drop the message on that. They will give you the recording. And in case recording of the 2 classes, cell this unit of life and the cell cycle and cell division. And even in case if you have any problem anywhere at any point, any problem, right? You can ask me in class, right? Even you can take the extra help, right? You can coordinate with those people, right? They'll arrange extra class for you, right? Okay, Aisha?
Yes, yes.
Fine. So what is the average? One more thing which I want to discuss with you. What is the average composition of the cell? And one more very important question. Iram, tell me. Lipid is macromolecule or micromolecule? That is the question. And you know, before answering me this question, I want to remind you that this question has been asked multiple times in medical examination, NEET exam. Yes, both of you. Even Aisha. Iram, tell me.
Micro.
Micro. How? So please note it down. Note. Lipids are considered to be macromolecule. Though they have molecular weight, less than 800 Dalton. So if we talk about the Dalton, if we talk about the size, so 18 to 800 they are considered micromolecule. Then lipid must be micromolecule, but yet lipid have the weight of only 800 Dalton, but still it is considered macromolecule. So this is the exception, right? So please note it down. Actually, full stop. Actually, lipids are not water soluble. We know. Actually, keep on writing. Actually, lipids are not water soluble. Full stop. They get separated in acid. Full stop. Otherwise, lipids are not. Otherwise, lipids are not strictly macromolecule. So because they get separated with the help of acid insoluble fraction, so that's why they are considered as macromolecule, which is the property of macromolecule. Otherwise, they are not strictly macromolecule. Okay. Next. Now, when we talk about the average composition of living cell, right? So what is the average composition of living? Average composition of living cell. Always jot down whatever I write in the whiteboard. So average composition of living cell. So what is the average composition of a living cell, right? So let me tell you. This sequence is it can be asked at your school as well in as a 1 marker question, right? So in average composition, the component of the water. So water is 70 to 90%. It depends. Plant cell, animal cell, fungal cell, or bacterial cell. What is it? Water. Then protein. Protein constitute 10 to 15%. 10% to 15%. Then carbohydrate means sugar. So carbohydrate constitute 3%. Then lipid constitute 2%. Nucleic acid constitute 5 to 7%. Ions 1%. Please write it down. This composition. Let me know when you note it down. Done?
Yes, sir.
Okay. Now, one more thing. You can see in page number 146 of your NCERT. So, primary and secondary metabolite. Can anybody of you, anyone of you, can tell me what is the primary or secondary metabolite? The next heading is, put the next heading, primary and secondary metabolite. What is the primary metabolite? What is the secondary? Look, the only difference between two primary and secondary metabolites is that the primary metabolite are those metabolites which are directly involved in the what? Which are directly involved in the chemical reactions, right? Or metabolic reaction. Means these are the chemical which are directly involved in the metabolic reaction of the cell, right? Secondary metabolite, they are not directly involved in the chemical reaction of the cell, right? Primary metabolites are basically those things without which the internal chemical processes, or we can say metabolic reactions, they cannot be completed. Means the main processes, main reactions of the cell will stop. So we can say all those things which takes part in respiration, photosynthesis, and all that, they are the primary metabolites. Second is secondary metabolites. Secondary metabolites are basically the additional compounds which are not directly involved in the basic main reactions, means the primary reactions or the metabolic reactions. They have some additional benefit, right? Like latex, they may provide the safety to any particular organism. They may provide to the any special peculiarity or a special quality to particular cell, right? These are called secondary. For sake of example, we can say that all those enzymes which are involved in the respiration or photosynthesis, they are primary metabolites. But many of the plants, they secrete latex for sake of what? Safety. For example, a rubber plant, whenever you pluck a leaf, whenever you pluck a leaf, it will start oozing out some latex kind of the substance. Same thing with the banyan tree, same thing with the peepal tree. So what happened? These are the milky substances. Basically, these are very highly, they have a bad smell, and like they do not have good taste. That's why the animal do not eat that, right? So these are the actually secondary metabolites. Many of the trees, when you get in touch with that trees or plant, you start getting itching, right? They secrete some chemicals, so which provide which impact uh, for the irritation or burning sensation in our body. So these are basically secondary metabolites. So the good oils which attract the or essential oils which attract the pollinator like bees and all that, they are the secondary. Did you got my point?
Yes, sir.
So please start writing now. First, put the heading metabolite. The organic compound like the organic compound like carbohydrate, the organic compound like carbohydrate, fat, carbohydrate, fat, protein, fat, protein, nucleic acid, etc. They are required for the basic metabolic process, of the cell, basic metabolic process of the cell throughout the plant kingdom. Full stop. These organic compound are called primary metabolites. Next point. The number of organic compound which are not involved in the primary metabolism, like photosynthesis, respiration, protein synthesis, etc. and seems to have no direct function in growth and development of plant. Full stop. Such compounds are called secondary metabolites. Now, what can be the question? Please, this is very important for your school examination. They can ask you as a 3-marker question, write down about the primary as secondary metabolite, and write down the secondary metabolite along with some of example. So you have to note it down. Some of the example of secondary metabolite, they are very important, right? And even this can be the 1-marker question in your examination. They can ask you which of the following is the secondary metabolite. So as I have already told you that these are the metabolites which do not involve in the directly in the basic metabolic reaction. So secondary metabolite. Let me write it down some example, and even you have to jot it down. Do not directly involve in growth, right? And the example are pigments, like color in plant, because they are for the attraction of attraction for the pollinator. So pigment and what else? Alkaloid. They are for the defense purpose, right? Essential oil for the pollination, to attract the honeybee and all that. Essential oil. Toxins. Some plant produce toxin as a defense mechanism, right? Drugs. They all are secondary metabolites. Is that clear, or you have any doubt? Note it down. Iram, Aisha.
Yes, sir.
Okay. Clear? Please note it down. 2 more point in secondary metabolite, because it can be asked in your school examination. Some of the secondary metabolite attract animal for pollination. And some of them help in defense against herbivore animal, because they can eat them. Herbivore animals, right? So this was primary and secondary metabolite. Biomolecule, micro and macromolecule, we have already discussed what are the macromolecule and micromolecule. Now we are going to discuss about put the heading macromolecules. So out of these macromolecule, we'll study protein, we'll study carbohydrate and all that, right? So start with the protein. In page number 147 of your NCERT, you can see, put your NCERT along with you, right? Give it to you, and we'll follow the sequence of the NCERT only, right? Whether the sequence is not that much lucid, but still. So put the heading protein. So can you tell me what is the monomer of protein? Next heading is protein. This is a bit complex chapter, but it's not that much complex. Just try to understand all the concept. It's not that much complex. You will get it every point at the end of the class. Protein. Iram, can you tell me?
Sir,
Aisha?
yes, sir.
Yes. What is the monomer of protein?
Homopolymer.
Homopolymer. Okay. Aisha, what is the monomer of protein? Means what is the constituent? I would like to make more clear my question. What are the basic constituent of the protein?
Polypeptides.
Okay, great. Aisha, any idea?
Glucose.
Glucose. They are the monomer of actually carbohydrate. So, look, let me tell you.
Lipids.
Lipids, they are a different biomolecule. Lipids are different biomolecule. Look, actually, protein are different biomolecule as we have discussed. Protein are different type of the biomolecule. So, type of the biomolecule: protein, carbohydrate, lipid, nucleic acid, and enzyme. They are basic 5 kind of the macromolecule or biomicromolecule which we are going to study. Right. So, first, it is given in our NCERT, that is a protein. Look, let me tell you. So basically, there are basic unit, or we can say monomer. So monomer. So monomer are the constituent or basic unit of any of the component, any of the thing in this world, right? Like we have some like a chain. So a single unit of the chain, that is a monomer, right? So monomer means basic. So there are some basic unit, these basic unit. So these basic unit will be called, they are single unit. So these each of the basic unit, they are called amino acid. Right? Amino acid. So this is the one amino acid, this is one amino acid, this is 2, this is 3, this is 4, this is 5, this is 6, this is 7, and so on. Right? These amino acid, they get arranged into one chain or many chain, right? So when these amino acid, they get arranged in a chain and they are connected by the bond. So there is a bond between these units, right? There is a bond between these units. There is a bond, right? So this bond, right? This bond, the bond which connect two amino acid, or the bond which connect two monomer, right? Two amino acid. So the bond between the two amino acid, this is the one amino acid, this is second, this is third, this is 4, this is 5, this is 6, this is 7, this is 8. The bond between, or the bridge between the two amino acid is called what? Can you tell me? Peptide bond. Right? So one amino acid remains connected with another amino acid with the help of a bond, or with the help of a link, we call it peptide bond, right? And what we call this chain? So amino acid get arranged in a chain and they make a polypeptide chain. Polypeptide. So amino acid get arranged into chain form, and this chain form is called polypeptide. Again, now this polypeptide chain or many polypeptide chain, they become folded accordingly, right? So this is the folded polypeptide chain. When this polypeptide chain get folded, it remains, these foldings remains connected with each other, right? And the thing which connect this folding, or the thing which hold this folding, they are called disulfide base. Right? So this folded polypeptide chain is called protein. Is that clear? Crystal clear? Tell me.
Yes, sir.
Crystal clear? Both of you? Aisha, is it clear?
Yes, sir.
Great. So just keep on noting down whatever I am telling you, because you won't find every diagram in your notebook, your book or helping book, right? So that's why I'm telling you. one more very basic kind of thing which I am going to tell you. Give me just one. I want to tell you the basic peptide bond. Peptide bond. Let me explain you more clearly what are the peptide bonds. Okay. So what is the chemical? Just first please note it down. Whatever visible to your screen, please note it down and let me know. After noting down, then I'll tell you. You can say this is the most complex chapter of complete 11th syllabus. No students want to study it because this is very boring kind of the thing, but you will get question in your school exam. It's not that much important for your medical examination, but it's very important for your school examination. After noting down, please let me know, then I'll go ahead.
Done.
Done. Fine. Okay. Now let me tell you what is the amino acid. So what is the formula of amino acid, or we can say what is the chemical composition of it. So there is a carbon in center. R means alkyl group, means CH2, CH3, CH2, CH2, CH3. This may be the hydrocarbon chain. Then there will be a functional group, carboxylic group, COOH, right? Then a hydrogen atom. Then this is hydrogen, nitrogen, NH. You can say it NH2, right? HNH, right? NH2. This is one amino acid. Is it clear? One amino acid. Clear?
So what is R?
R means alkyl group. We always denote in chemistry alkyl group. You must have listened. R means that is like CH2, 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 CH3. It may be a long chain of hydrocarbon. Clear?
Yes, sir.
So this is one amino acid, right? This is. Let me make it more clear. This is one amino acid. It is the generic formula, right? It is the generic formula of amino acid. Clear? It's a general formula of amino acid. Now there is another. There will be another amino acid. So again same C. Again there will be COOH. Again, what else? That is H N H. What will be here? Again R alkyl group. And what will be here? Again there is a hydrogen. Now what will be the peptide bond? So when hydroxyl group of one amino acid and hydrogen of another amino acid, they combine, and what they will make? They will make water molecule. Yes, is it clear? H2O. They will make H2O. The H2O will be removed, and the formation of peptide bond between these two will take place, right? Then the formation, then what will happen? Then again, the formation of peptide bond between these two will take place. So what will happen? H and H, carbon, alkyl group, and here CO, and here H. What will happen with the next one? Then it have lose hydrogen, so NH, again C, R, COOH, and then H. So this will be called this one will be called peptide. This is the peptide. Is it clear or not?
Yes, sir.
And whenever two amino acid will combine, they will lose. Whenever two amino acid will combine, they will lose one water molecule, and formation of peptide bond will take place. So CONH is a peptide. Yes, please tell me.
So HNH and HNH do not combine together. Like instead of COOH, HNH do not combine together.
No. They just lose the word like NH, NH2. This is H and H. We can call it NH2, right?
NH2, yeah.
NH2 means amide means we write it in this way. NH, NH. Correct? HNH. Right. So what? It loses its hydrogen ion, right? And it loses its hydroxyl. And both combine, they makes water, right? So whenever two amino acids combine, they loses a water molecule and bond between CO and NH takes place. This is called peptide bond. Is it clear? Iram, is that clear?
Yes, sir. Clear.
Aisha,
Yes, sir.
please write it down. This is very, very, very important. If you got it, it means you got the this one, biomolecule check.
Done.
Done? Okay. And clear to? Clear to both of you? Okay. So please start writing now. Proteins are the large sized, proteins are the large sized macromolecule. Proteins are the large sized macromolecule having one or more polypeptide chain, one or more polypeptide chain. That's what I was telling you, polypeptide chain. So in one protein, there may be one polypeptide chain or more than one polypeptide chain, right? Full stop. The term polypeptide, often used for protein. The term polypeptide often used for protein. Full stop. Next one. A single polypeptide chain must be 50 amino acid, 50 amino acid longer, 50 amino acid longer to qualify this term. To qualify this term. Is it clear? Did you got this point?
Yes, sir.
If a chain of amino acid is 50 or more than 50 amino acid longer, then only we can say it polypeptide chain. If it have 49, 48 or 45 amino acid, we cannot call it polypeptide chain, right? Next, very important, underline this next point. They are the most abundant, and most varied. They are the most abundant and most varied macromolecule and most varied macromolecules, as they constitute about 50%. They constitute about 50% dry weight of cell. They constitute about 50% dry weight of cell. Next point, even next point will be very important. Underline the next point. Even
So what does dry weight mean?
dry weight means, Iram, we have to remove the water. Like suppose that my weight is 70 kg, right? So 70 kg is not dry weight. 70 kg is my weight. For example, you have spinach or any of the vegetable, right? You have a carrot, 1 kilogram carrot. That is a weight. What you have to do, put it in a machine. The machine which dehydrate complete water, and if you will remove the water from the carrot, it will become hardly 250 gram or 200 gram, right? So that is called dry weight. Clear?
Yes, sir.
So that is dry weight. So next point is a question. It has been asked multiple times in NEET examination. Closely related species have similar type of the protein. Closely related species have similar type of the protein, while distinct type of the species have uncommon protein. Means the protein of chimpanzee and human will be very close, and the protein of human and deer will not be close, right? So if proteins are matching with any species, means they are closely related. The species is close to the human. Got it? Got my point?
Yes, sir.
Next question. Next two points are the questions which have been asked in medical examination multiple times, right? Can you tell me what is the most abundant protein in animal? I'll tell you. Leather is made up of that protein. Can you tell me? Our skin is made up of that protein. Animal skin is made up of that protein. Huh? Which one?
Collagen.
Excellent. Yes, collagen. Very good. Because leather is made up of collagen protein. So please note it down. Collagen is COLLAGEN. Collagen is the most abundant protein in animal world. Collagen is the most abundant protein in animal world. Can you tell me the most abundant protein in plant world? And what is that? These are all questions of medical exam. 100 of time they have been asked. They are older questions. Previous year questions. There is an enzyme we will discuss, the plant enzyme. That is the most abundant enzyme of the earth as well, right? and that is the most abundant protein of the earth as well, right? The name of that enzyme is rubisco. Rubisco is a plant enzyme, right? It stands for ribulose biphosphate carboxylase oxygenase. Let me tell you. Ribulose biphosphate carboxylase oxygenase.
So,
Yeah.
sir, abundant protein do not perform any functions.
Abundant protein do not
perform any functions. That's why they're known as abundant.
No. Abundant means they are the most wider spread. They are found in larger quantity.
Okay. Yeah.
Right. So means if someone asks you, tell me the name of the protein which is found in the largest amount on the earth, means the most abundant enzyme, right? The most abundant protein, right? So the most abundant protein is rubisco in plant food, right? So please note it down. Rubisco is not only the most abundant protein in plant. Rubisco is not only the most abundant protein in plant, but the whole earth. Means if you get the question, tell me the most abundant protein of animal world, then the answer will be collagen. If I ask you question, tell me the most abundant or the most wider spread or which of the protein is the found in highest quantity in the world, then you will tell rubisco. So most abundant protein of the earth, rubisco. Most abundant protein of the plant, rubisco. Most abundant protein of the animal world is collagen. Is it clear?
Yes, sir.
Means rubisco is found in more than animals because the quantity of plant is more on the earth than animal. Yes, clear? Aisha?
Yeah.
Aisha?
Yes.
Okay, fine. Next, the protein may be, so protein may be of three types, right? On the basis of their structure. Types of protein. So next heading is types of protein. Now come to the type of protein. Please note it down. On the basis of structure, protein may organize itself. Protein may organize itself in four levels. The first level is primary protein. So what is the primary protein? Simple polypeptide chain is called primary protein. You know, do you have any idea that when you egg is liquid form, but when you drop the egg or you put the egg white or yellow part in any fry pan and increase the temperature, why it becomes solid? Any idea? Any idea? No. You know, when it is a raw protein in egg form, this is this raw protein in egg form. This is like tertiary structure, right? Tertiary structure. And when you put it in fry pan, the tertiary structure means bond start breaking down, right? and tertiary structure become primary structure. So tertiary structure is in liquid form. When the structure changes to the primary structure bond due to breakage of the bond, it becomes solid. That's why omelette becomes solid when it is put in egg becomes solid when it is put in a fry pan. Is it clear? Did you got my point? That is only change in the structure of protein. Okay. So please, what is the primary structure? Primary structure is that like it is a polypeptide chain, like this kind of polypeptide, right? And the distance between two adjacent polypeptide bond means the distance between a polypeptide bond is 0.3 nanometer, 0.35 nanometer. So if the distance between the two adjacent peptide bond is about 0.35 nanometer, then and a simple chain is called primary structure. Make this a structure and note it down. This is the basic structure of protein. This is the basic structure of protein. Let's stop. The distance between two adjacent, peptide bond is about peptide bond is about 0.35 nanometer. Right? Now the secondary protein. The second level of organization is
Sir, how long is this chain? How long?
there is no limit. It may be of any length. But this will be single chain. Secondary structure. Now, what about the secondary structure? Secondary structure, there will be more folding, right? So, for sake of example, it may be like this way. Suppose that this kind of the helical structure, or you can say there may be this one polypeptide chain, right? Another polypeptide chain. And I have already told you that the polypeptide chains remains connected with each other with the help of a bond, and we call it disulfide bond, right? So there will be bond between. Here will be a bond. This will be a bond. There will be a bond. So these structures are supported by bonds, right? And these bonds are either called hydrogen bond. They remains connected with each other with the hydrogen bond and disulfide bases. These are the basically hydrogen bond. So they remains connected with hydrogen bond. And this kind of the structure, this helical structure, this is called alpha helix. We call it alpha helix. And this structure is like sheet. So this is called beta plated sheet. Beta plated sheet. So in secondary structure, what happened? Many polypeptide chains, they get arranged in two forms, either in alpha helix or in beta plated. And these polypeptide chains remains connected with each other with the help of hydrogen bond. Is that clear? Please start noting now. The amino acid present in linear sequence. The amino acid present in linear sequence become more complex. Full stop. They get arranged. They get arranged in the form of alpha helix, comma, beta plated sheet, and collagen fiber. Full stop. Two or more polypeptide chain. Two or more polypeptide chain coil around each other or get arranged in the form of sheet. Full stop. Is that clear? Now come to the third one. So the third one is tertiary structure. Now further folding will be tertiary structure. Tertiary structure will be more folded, right? Again, this is a structure where these are the hydrogen bond. Hydrogen bond. Along with hydrogen bond, there will be some more things like disulfide bridges. Disulfide bridge. Or you can say sulfur-tron, right? So please note it down. When secondary structure folded down further, then they become tertiary structure, right? So this is nothing but more complexity of the folding, or this is the more like complex folding, nothing else. Start noting down. There is bending and folding of various type of form sphere rods form sphere rods or fiber. Full stop. Tertiary structure is stabilized by several type of bonds. Tertiary structure is stabilized by several type of bonds like hydrogen bond, ionic bond, van der Waal bond, covalent bond, etc. Right. last but not the least, that is the quaternary structure. Quaternary structure. The best example of the quaternary structure is like hemoglobin. Quaternary structure. I have told you like the quaternary structure, that is the hemoglobin. Hemoglobin. It is made up of more than one type of the. This is made up of more than one type of the polypeptide chain. Right? Hemoglobin is made up of 4 polypeptides. 4 polypeptide. Out of them, 2 are alpha and 2 are beta. Two alpha chain and 2 beta. Right. So please start noting now. This is found only in complex protein like hemoglobin, full stop. The different subunit chain, fit together to form a complex structure. Now tell me, is that clear? Along with the class, go through with your NCERT and please tell me. Is that clear, or you have any? Do you have any doubt? Both
No,
of you. Okay. What about you, Aisha?
No, sir.
Everything clear?
Yes.
Okay, fine. Now the second thing, second heading is in your NCERT, page number 148. That is a polysaccharide, right? So we have already discussed that. What happened? Oh, we have already discussed that the first macromolecule was protein. Now the second macromolecule is carbohydrate, means sugar. Right. The second one is that is the carbohydrate or polysaccharide. So polysaccharide, carbohydrate, both are the same thing. Polysaccharide. Polysaccharides. So polysaccharides and sugar or carbohydrate are synonymous to each other. You can say carbohydrate, or even you can say it's sugar. They all are the similar term, right? All are the similar term. Now, in the same way, as we have already discussed with you, right? Try to understand one by one each and everything, right? What is the polysaccharide? What is sugar? What are the carbohydrate? Right. So carbohydrate. So can any one of you tell me what is the monomer of carbohydrate? Tell me. Look, there are a very basic kind of unit What are those basic units? Monomer? Glucose, come on. Glucose. So when these glucose, so glucose is the most basic component, or glucose is the monomer, right? Monomer. When number of the monomer combine and they get arranged in a chain, they are called polymer, right? So number of monomer get arranged in a chain. Right. So when glucose combine with each other, and what we call the bond between these two glucose molecules. Can you tell me? Yes, guys. Two carbohydrates get arranged themselves in a bond. What we call that bond? So the bond between two amino acid peptide bond, and bond between the two glucose molecule is called glycosidic bond. Glycosidic bond. Glycosidic bond, right? So glycosidic bond is found between the two sugar molecules. So they get arranged. So when the number of glucose molecule get arranged in a sequence with the help of glycosidic bond, we call it a polysaccharide. This chain is called polysaccharide, and the polysaccharide chain is called what? Sugar. Sugar may be sweet or may not be sweet. It is not necessary that every sugar is sweet, right? Sugar may be sweet, may not be sweet. Is it clear?
Yes, sir.
Yes.
Please note it down. This is structure. One more thing which I want to tell you. For example, this is one sugar. This is a structure of one glucose molecule, right? And here it will be hydrogen. Here it will be OH, right? And again, another glucose. These combine, and again the water molecule is removed. When water molecule is removed, and the bond between, after the removal of the water molecule, the bond between these two. So what kind of the bond formation will take place? So what kind of the bond formation takes place? The bond between these two sugar molecules, this bond. This is called actually glycosidic. So the bond between two amino acid is called polypeptide bond. The bond between the two sugar molecule is called glycosidic. Please note it down. This is structure. And next class we will start with the carbohydrate. And till the time, please go through with whatever we have discussed today's class, and tell me if you have any doubt regarding that. Tell me when you note it down.